Well, first of all, good afternoon, everybody, and thanks to ERGA for selecting Dysdera unguimmanis to be sequenced, and also for offering me the opportunity to talk about this species. So I'm Nuria Macías Hernández from the University of La Laguna, and this project is being made with the collaboration of other colleagues from the other universities from Spain and Europe. So as you can see here, Dysdera unguimmanis is a cave dweller spider that inhabits the island of Tenerife. In this map, you can see the distribution of the whole genus, the genus Dysdera, that is distributed in the whole Mediterranean basin. It has like more than 300 species, but is in the easternmost distribution in the Canary Island, where this genus has undergone an adaptive radiation with more than 60 endemic species. One of its endemic species is Dysdera unguimmanis, that it inhabits 2 caves in the northern slope of Tenerife. Next, please. So Dysdera unguimmanis is the most troglodytic species of the whole genus. Down in the picture, you can see an epiphysean species, also an endemic from Tenerife that is darker with shorter legs, in comparison to Dysdera unguimmanis. As you can see, that is depigmented with very elongated legs. It has no eyes, so it's completely blind, so perfectly adapted to the underground environment. Indeed, the genus Dysdera is a perfect model for studying the evolution of cave life. In the case of the Canary Islands, 10 of the 60 endemic species are troglodytic. And thanks to previous phylogenetic analysis, we know that these species have colonized the underground environment in 8 independent occasions. Finally, these species have very restricted distribution. So previous IUCN assessment suggested that these species could be potentially endangered by local threats and global climate change. Next, please. So there are several species, epiphysean and troglodytic species of Dysdera, of the family, that are now being sequenced. So in the future, with these genomes available, we will be able to compare several genomic studies to understand the evolution of the adaptation to subterranean habitats, as well as the genomic basis behind adaptive radiation and speciation. And finally, knowing with all this comparative genome analysis to understand how these species that has very narrow ecological niche are adapting, which responses are they adapting to environmental stress, like, for example, temperature or climate change. Well, thank you very much.